Hi, good evening. So today we'll be looking at the nervous system and then its functions and rules when it comes to the activities of the whole body. So when we take the human body is basically a design just like a car, which has an electrical wiring system or a steering system, which is the nervous system. It has an engine system with some vessels, which we call the heart and the cardiovascular system. It also has a breathing system, which we call the respiratory system. So we are going to look at the steering part of or the electrical aspect of or the senses of the car, that is the nervous system. So basically the nervous system is divided into two main parts. We have the central nervous system, which is located at the midline, which is made up of the brain and then the spinal cord. And then the peripheral nervous system, which is made up of the nerves that are connected to the brain, which are known as the cranial nerves, and those that are connected to the spinal cord, which are known as the spinal nerves. So the peripheral nervous system will be made up of the nerves and the ganglia system. So the ganglia are just collection of cell bodies that we found in the peripheral nervous system, while nerves will be collection of the axons. So these nerves that we see here can either be the nerves that we see here, can either be taking information to the central nervous system, to the brain for processing, for interpretation, or they may be the brain may give a command through this nerves, or there could be an interplay between this nerve to this nerve. So what happens is that when those that take the information to the central nervous system are known as the sensory nerve, so they do the sensory input. And when the brain gives a command, those that take the information to the muscle or the organs to function are the motor output. Just like a computer, you key in you give your input through the keyboard or the mouse system, and then you have the output on the screen. The integration system is going to be like the monitor and those that to process the work. So that is what we are talking about in this section. So when we look at the general organization of the nervous system, so the central nervous system, which is made up of the brain and the spinal cord, will have a somatic, a special sensory receptors and neurons that are connected to it. So when we say somatic, we means body. So that means that we are looking at receptors that to convey body information or special sensory receptors such as eye, taking information for vision, the ear taking the information of what audition and all those things to the central nervous system for interpretation. But then autonomic sensory nervous system can also relate with the central nervous system, where we look at the information that goes to the body organs, such as the visceral organs, such as the kidney, the intestines, the lungs, and all those things. So the peripheral nervous system, which are taking the information, can eventually be divided into three main parts. The somatic nervous system, the autonomic nervous system. Then we also have a special nervous system that we call the enteric nervous system, which play a role of information to the GIT, that is the gastrointestinal tract, and has a lot of enteric plexus. So from the central nervous system, a somatic and special sensory information should be conveyed via somatic motor neuron, which are usually voluntary, And then the information, the effect is seen on the skeletal muscles. Now, when we take the autonomic motor nervous system, the effect is classified into two. Either it is a sympathetic effect or a parasympathetic effect. A sympathetic effect is an effect that will allow for agitation of the body, or when the body is agitated. And then parasympathetic will play a role when the body is in what? Rest mode. For instance, you have finished eating 5 balls of banku, and then what you do now is that you relax. As you relax, whatever is happening is you are in the parasympathetic state. But for instance, if you want to get into a fight, or you got into a fight, the part of autonomic nervous system that is responsible for the action that we see on the heart, increase in heart, your eyes are widely open, and all those things, you begin to your heart beat faster. All these things are termed as a sympathetic, and you see these effects on the smooth cardiac and the glands. Then this autonomic nervous system has an interplay with the enteric motor neurons, which convey information to the motor smooth muscles, the glands within the GIT, and then the endocrine cells of the GIT. So this is the total organization of the nervous system. This is what I was trying to explain earlier in the earlier lecture, in the earlier slide. So now let us continue and look at. So basically, the nervous system is made up of the neuron, a neuron. Now, a neuron is the basic unit, or is a basic functional unit of the nervous system. And this neuron comes in different forms. So usually the part of the neuron, the neuron will have a cell body, which is the processing center of what? The cell of the cell. So this is a whole one cell, okay? This whole thing is a cell. It has a cell body where you have the nucleus, and then you have the neurofibril with the dendrites attached to it. Then one of the extensions of the cell body is the axon. So what you have to know is that there's a cell body and there are extensions on the cell body. Some of the extensions are shorter and they form the input part, which we call the dendrites. And the one is a longer, which forms the output part, which we call the axon. So at the axonal part of the neuron, we could have branches which we call axonal collateral. Then there could be some glial cells that are on it. Glial cells are the supporting cells, as we know earlier. So this is a Schwann cell kind of what wrapping around the axon. We'll be seeing the function of this later. So the Schwann cell also has its own cytoplasm, and this Schwann cell actually forms something we call the myelin, which kind of insulates, cause some form of insulation to the cell. Now, the space between the myelin, where we do not have any between the Schwann cells, where there is no myelination, is what we call the node of Ranvier. The nodes of Ranvier. So this is just to speak about, show us the neuron. So neurons come in different formats. Okay, the very first thing is that before we look at the different types of neurons, as I said, these are the receiving ends of the neuron, so they serve as the input. This is the integration part, and then this is what is going to give the output. So what happens is that when there is an impulse coming into the cell body, it is picked by the dendrons, get into the cell body where there is integration. Then there is going to be what? A spike initiation of that impulse, which is going to be relayed to the axon terminal. And this axon terminal is where the neurotransmitters are released to give information to the effector. One thing I would want you to find is what is the essence of the nasal bodies and the neurofibril, and what is the differences between the two. So axons can communicate with another axon, and when they do that, we call them synapses. So a synapse is actually a junction between two neurons. So when two neurons are kind of communicating, sorry, as I said, axons will communicate with the cell body, not usually an axon. It can communicate with any part of the neuron. So when this communication comes and the axon is communicating with the next cell body, then we call that what? Um, axodendritic synapse. So the synapse can be axodendritic, where an axon synapse is a dendrite. And then the axon can also synapse with what? The cell body, where we call that the axosomatic. So this is an example of axodendritic, and then this is an example of axosomatic. Then we can also have the exoaxonic synapse or axoaxonic synapse, where an axon communicates with another axon. So usually, it is not just one type of neuron that convey similar informations in the nervous system. So neurons can actually be classified based on their shape. So let's go to the white body, and then we'll look at the classifications of neurons. So neurons, sorry. So neurons can be classified. When we take the neuron, we want to look at the classification. We can classify neurons based on shape. So based on shape, we could have stellate neurons, could have basket neurons, could have pyramidal neurons, we could have Purkinje neurons. Then we could also have, based on number of extensions on the cell body, we could have one unipolar, which looks like this, just one extension on the cell body. We could have bipolar, which has two extensions, so an axon and a dendrite. And then we could have multipolar, which has an uncountable dendrite system, plenty, and usually with one axon. So that is based on what? Number of extension. We can also classify these same neurons based on their function. And based on their function, we can call them what? One, sensory neurons, relay neurons, and motor neurons. Sensory neurons, relay neurons, or motor neurons. So these are some bit of classifications when it comes to classifying neurons. Nonetheless, milk concentration is going to be in the number of extension paths. So let's go back to the slides and then we continue from there. So just I was saying, this is the different types of neuron. However, there is also another neuron which looks like it is unipolar, but it is actually not coming. It's not a unipolar. Which looks like it is a unipolar, but actually it gives a false impression that it is a bipolar neuron. So when you look at this neuron is actually called pseudo-unipolar. Pseudo-unipolar because it comes down and then branches into several parts. So this is a pseudo-unipolar neuron. I've also talked based on the structure or based on the shape. So we could see Purkinje cells as you see in the cerebellum, pyramidal cells as you see in the cerebrum, which are all multipolar. Then we can also look at the olfactory cells or the retinal cells, which are usually bipolar. And then we can look at the dorsal root ganglionic parts, which are usually unipolar or pseudo-unipolar. We could take time and then we'll look at the various types of neurons and then the activities they do. So this is just to tell us the differences, the comparison between based on polarity, the types. So usually the multipolar neurons are motor in function, while the bipolar and the unipolar are usually sensory neuron. At times we could also have some of them be motor. And so most unipolar neurons, multipolar neurons, we know are motor neurons. That is one thing we have to know. And most multipolar neurons are basically motor neurons. So just continuing with neuron types, we can have a look at this and then we could make research on them and know why they are looking like that and where actually we can find them. Now let's come to the supporting system for the central nervous system or for the nerves. So usually the neurons are actually supported. They have a supporting neuronal system which we call the glia or neuroglia. So neuroglia or the glia are all supporting cell systems for the neurons. So the glial neurons are very essential. They actually kind of take care of the main neurons. And there are 6 basic types. 4 of them are in the central nervous system, while 2 are in the peripheral nervous system. So astrocytes, which we usually find in the central nervous system are associated with the formation of the choroid plexus. And then we are also looking at the microglia, which function as a kind of a defense system for the nerves. So the astrocytes, which will form the blood-brain barrier, not the choroid plexus, sorry, the blood-brain barrier, where there is a barrier that is formed between the blood that is coming to the brain and then the brain substance itself. So that is done by the astrocytes. The microglia are usually defensive in action, and they perform that defense by phagocytic activities. And then oligodendrocytes perform the same function just like the Schwann cells in the PNS by forming of what myelin sheaths around the axons. Then the ependymal cells, we can see them lining the ventricles, and then they are kind of also attached in the formation of some form of choroid plexus, and then leading to the production of cerebrospinal fluid. So these are the ones that we see in the central nervous system. So histologically, the astrocytes, which form blood-brain barrier, looks like this. The oligodendrocytes, sorry, they are the myelin-forming cells, and they can actually myelinate. One cell can actually myelinate more than one axon. These are the microglia, which are phagocytic in function, and then the ependymal cell, which is involved in the formation of some form of choroid plexus for the production of CSF. What we have to note, as I said, is Schwann cells, I've already said, is myelinate a single axon, whilst oligodendrocytes myelinate a lot of axons. And these myelin sheaths, so this is an axon, this is a Schwann cell, which is either produced by oligodendrocytes or the Schwann cells. So these are the myelin sheaths, and then this we can see one myelin to the other. We can say that this is Schwann cell. But when you can see one cell and then a lot of nerves inside, then we can call that the oligodendrocytes. Okay, so when you look at this, this is actually myelin Schwann cells producing myelin that will kind of insulate the axon. But here we can see that the axons are unmyelinated. You cannot see any myelin covering the axons. Okay, you cannot see any myelin that is formed. So usually that is why we don't have a lot of myelination when it comes to the brain. But when it, or the central nervous system, but for the peripheral nervous system, you have a lot of myelin formation. Now, the gaps that we have between the Schwann cells or the myelin sheath is what we call the nodes of Ranvier. And these nodes of Ranvier are also actually involved in the signal transduction across the cell membrane. So the last of all the glial cells is the satellite cells, which is also seen in the peripheral nervous system. And they actually provide more of support and regulates ion inflow and out of the cell. So one beautiful thing is that this myelin sheath that we see in the peripheral nervous system, when there is a breakage in or there is a damage to the axon, this myelin sheath can actually guide the regrowth of this axon. So let's say we take this place, see that this whole place is broken. Can say fragmented axon, droplets of myelin sheath. They can actually guide, this is the site of the damage, guide the formation of what? The axon. So eventually you are going to have something like this, and then you have this one. So the very first one is that when there's a damage to the cell, what happens is that the macrophages come in to serve as what? A defense system. As they do that, then there's going to be a directional flow for alignment of the nerves. So fine axons will sprout or filament to sprout to join each other. And then the axons will also align, forming the myelin around them. So aligning Schwann cells from regeneration tube will form so that they could cover this. And eventually you have your axon, and then the site where new myelin is formed. When you come to the brain, what happens is that this is spinal cord and this is brain. So usually the spinal cord has an interior gray matter which looks like H, but the brain has an exterior gray matter that we can see, and also another inner gray matter which is interspersed by what? A white matter. Now the gray matter are collection of cell bodies which are and unmyelinated axons, dendrites, and glial cells, while the white matter consists of myelinated axons. So the gray matter will consist of cell bodies, cluster of cell bodies, unmyelinated axons, dendrites, and glial cells, whilst the myelinated axons will appear whitish. And you must know that the white matter are always axons, but the gray matter can be cell body, it can be a glass cell, or it can also be a myelinated axon, unmyelinated axon, and dendrite. So we are going to look at how information travels to an effector. Here, the effector can be a muscle, it can be a gland for it to work. So what we have to note is that neurons will receive input from another neuron, especially through dendrites. So we look at when we look at the synapses that are formed, we look at the axoexonic, axosomatic, and axodendritic interaction. So when you look at this, we are going to see that we are having an interaction between the soma and then this dendrite. So this is these two neurons. So neurons will always send their output in the form of an action potential. So what is an action potential? We discussed this when we were looking at muscle physiology. Now, when an action potential arrives at a synaptic end bulb, there is going to be a neurotransmitter that is going to be released, as we saw when we were dealing with muscle physiology. This neurotransmitter will bind on receptors on the postsynaptic neuron, which often opens ion channels. So the flow of ions cause an electrical current in the membrane. So what we are trying to say is that, let's go to the whiteboard so that we look at the demonstration. So assuming we are looking at the somatic nervous system, where information is just coming from a neuron, let's say one motor neuron with plenty dendrite and one axon. And then these are the axonal ends. And then this is, let's say, the muscle. Here, we are having what we call one neuron. So this is the synaptic cleft. This is the synaptic cleft, the gap between. That's the synaptic cleft. Now, when action potential travels and reach here, neurotransmitters are formed in vesicles, which are released, especially acetylcholine. So when acetylcholine is released, it binds to receptors on the effector, which causes the opening of gated ion channels, causing a depolarization in that particular effector membrane, hence will cause the activity of that membrane to take place. Now, what we have to notice that when we look at this, the neuron that is giving the information to the is known as the presynaptic neuron. So we can have a neuron that is presynaptic. So this is the somatic nervous system, where we have only one nerve that is actually one long nerve that is conveying the information to the body system. And somatic could be the muscle, the body, and the skin. So this is the presynaptic neuron, which releases a neurotransmitter. Now, when it comes to the autonomic nervous system, it consists of what? Two types of what? Neurons that convey the information. Two arrays of neurons that convey the information to an effector. So you could have the first one, which is a bit shorter, and they have a longer one. And then we could also have one which is longer, and then we have a shorter one. So what happens is that in the autonomic nervous system, these are the synapses that are formed here. So there's a synapse. So the neuron here is known as the presynaptic neuron, and the neuron here is the postsynaptic neuron. The same thing applying to the neurons of the So one design, I nearly said it. I want you to go and find which design where we have short preganglion synaptic neuron and long presynaptic or ganglionic neuron, and then long preganglionic neuron and short postganglionic neuron. Which of them fits for what type of the autonomic nervous system? So what you have to know is that for one of the nervous system, which is a sympathetic, okay, let me clear this. So when you take, let's say, the arrays of neurons, if it is, let's say this is the autonomic nervous system, and we are looking at sympathetic nervous system, what happens is that there is a first neurotransmitter that is released, usually epinephrine or noradrenaline. This will have an interaction with the cell body or the axons on the cell bodies, which is going to cause processing over here, and then an action potential travels along this axon. And when it gets here, another neurotransmitter, which is usually acetylcholine, is really think of changed it. So usually what happens is that the presynaptic neuron will always release an acetylcholine over here. And then the postsynaptic neuron, depending on the effect, if it is sympathetic, will release epinephrine or noradrenaline. And if it is parasympathetic, will continue to release the acetylcholine. So the neuron here is the presynaptic neuron, and the neuron here is the postsynaptic neuron. So depending on the activity in the autonomic nervous system, we could have what? An epinephrine or an acetylcholine release from the postsynaptic neuron before it binds to an effector that is here, which could be a muscle, a smooth muscle of the intestines, or any glands that we see. So this is the array of what nerves and their signal transduction to the body entirely. Let's go back to the slides and see what is there. Now, as I said, the flow of ions will actually cause an electrical current in the membrane. That is the depolarization. Then these depolarizations can either be a positive or depolarization or excitatory effect, or you could have a negative, which could be a potential, which could be hyperpolarization potential, or which has an inhibitory effect. Now, one thing we have to know that the graded potential are summed in the axon hillock. They are summed in the axon hillock, like the neck region. And if the sum exceeds the threshold, then the postsynaptic neuron will fire an action potential. If the sum exceeds the threshold, the postsynaptic neuron will fire an action potential. When the action potential reaches the synaptic end bulb, neurotransmitters are released, and then the cycle begins again, which I explained on the whiteboard. When you look at this, we have a sodium gated channel that is closed. So at point 1, this is the threshold of excitation. So for the information to overcome the threshold, the sodium channels will open, and then this will cause sodium to enter and then potassium going out. This is going to cause a distortion in the membrane, leading to what? Depolarization of the membrane. And then as potassium begin to leave the cell, you have more depolarization of the membrane. Then at point 3, we call that a refractory period. Yeah, sodium channels become refractory, no more. That means they are so much that we cannot have more coming in. So we call that what? The refractory period. Then potassium ions continue to leave the cell, causing membrane potential to return to its resting level. The potassium channel will close, sodium channels will also reset, and then the whole thing begins. So this is just a pictorial demonstration to show how sodium ions are entering into the cell and potassium ions are leaving the cell. So action potentials, we say, it can be graded or local. So one thing we have to know is that until the threshold is met, action potential doesn't really take place. So this one, the black ones that we see here are the resting membrane potential. And when there is hyperpolarization, you can see this thing will begin to go down. the resting membrane potential. The membrane potential is measured in millivolts. So you see that charge coming down. But if it is depolarization, you see the charge actually going up. So it will be becoming more positive. But in the hyperpolarization stage, the membrane potential will be becoming more positive, negative, more negative. Whilst in the other one, the polarization graded potential, you are going to see the membrane to be more positive. One thing, we say the signals are graded because they vary in amplitude depending on the strength of the stimulus, and the signals can also be localized. So we want to look at the difference between the potentials of excitatory and inhibitory signals. So as we said, for excitatory, what will happen is that there is going to be a resting membrane potential where there is an action potential which causes a rise, and then eventually there is a decrease down, and then a rise back into its membrane potential. For the inhibitory signal, what we see is that there is a decrease in the membrane potential of the cell. Now, let us look at what actually triggers or the action potential or the threshold zone. So what we have to note is that the graded potential that arise in the dendrites converge in the axon hillock, as I said earlier. So when they get to the axon hillock, and then the sum of this potential is more than the threshold, then it's going to give an excitatory effect. But if the sum is lower than the threshold, then it will give an inhibitory effect. So that's what we are saying here, that if the sum exceeds the threshold value of the neuron, usually about -55 millivolt, then an action potential arises in the trigger zone. So unlike graded potential, the action potential is all or none. So until we have the information reaching the axon hillock here, we call them graded potential. When they reach the axon and then the sum passes the threshold, then we call that the action potential. So action potential can be all or none. Either it is giving an excitatory, or it is not doing anything. So when an action potential arises, it propagates along the axon, and when it reaches axon terminal, it may synapse with another axon, which could be the postsynaptic neuron, the postganglionic neuron, and could cause another graded potential in that neuron. And when it reaches, it's also the axonal hillock, then it could cause an action potential to also propagate along that neuron and then cause a release of a neurotransmitter from the postsynaptic neuron, which could be, if in the autonomic nervous system, could be sympathetic or parasympathetic. Now, let us see what can influence the speed of the impulse propagation. One thing we have to know is that the speed of an impulse is not related to the stimulus strength. One thing we have to note is that larger fibers will conduct impulse faster due to their size. For instance, when you take a dual road, cars are able to move freely on that dual road as compared to a one-lane road. And then myelinated fibers also conduct impulse faster due to saltatory conduction. Saltatory conduction is when there is a leaping form of the impulse, when the impulse decides to jump from one node of Ranvier to the under node of Ranvier. So in that case, the impulse is not actually transferred along the whole axon, but it jumps. So depending on the size, we are saying that we have delta fiber, A fibers, B fibers, and C fibers. And A fibers are the biggest, and they're actually myelinated somatic sensory or motor axons that gives an effect to the skeletal muscle or takes their information from the skeletal or the skin. Then the delta B fibers are medium, and then they actually myelinated visceral. So they are actually conveying information which could be which is usually sensory or autonomic to the autonomic nervous system. Then the delta C fibers, which are the smallest, are usually unmyelinated. So if we realize delta A and B will usually be myelinated, but C will be unmyelinated, and they're actually more sensory in function, and they could also be autonomic motor. So let's look at the conduction method. We earlier heard of sortatory conduction. So let's look at the two types of conduction. We are looking at the continuous conduction and sortatory conduction. So continuous conduction is usually seen in unmyelinated axon, where there is a step-by-step depolarization of each portion of the axolemma, that is the membrane of the axon. Then sortatory conduction is actually seen in myelinated axons, where depolarization only occurs at the nodes of Ranvier, where we have a high density of voltage-gated ion channels and cause a leaping mechanism of conduction from one myelin sheath to the other myelin sheath. So this is just to show where the sodium voltage-gated channels are actually more where we can find them more in the axon. So because of that, this is going to jump all the way to here instead of propagating along the axons before reaching here. That makes the impulse transduction or conduction faster. So let's look at the story so far about the nervous system and its transmission. The one thing we have to know is that an action potential will propagate over the surface of an axolemma, and then sodium will flow into the cell, causing a dramatic depolarization. So in response to this depolarization, voltage-gated channels, that is for potassium and sodium, will open. And when this happens, they're also causing some form of what? Propagation along the membrane, causing the stimulus to travel. Now, potassium flow out of the cell will cause a dramatic hyperpolarization. And then the resting potential of the membrane is gradually restored following a refractory period. So the action potential that travels along the axolemma is known as a nerve impulse. And when the nerve impulse reaches the synaptic end bulb, a neurotransmitter is released. And then the neurotransmitters will bind to their receptors on the dendrites or soma of the postsynaptic neuron, and then they produce graded potential. The graded potential are summed in the axon hillock, and if the sum exceeds the threshold, then an action potential is produced. So this gives us the difference between the overall summary on nerve transmission. So we want to see a comparison between graded and action potential. So graded potentials we see them around the dendrites and the cell bodies. and action potential are only at the trigger zone of the axon highlights. And then action potential is produced by voltage-gated ion channels, whilst GP is produced by ligand or mechanical gated channels. GPs are usually localized. They do not move. They only occur within a short path like the cell body. But the APs they propagate over the surface of axon, conveying the information from one neuron to the other. The amplitude of the action potential is constant, whether all or none. However, gradient potential vary depending upon the stimulus strength. The action potential is always the same, and however, the duration of the GPs is as long as the stimulus lasts. So action potential always moves as fast as it needs. But for gradient potential, the longer the stimulus, the longer they'll stay there. So this brings us to the end of nerve physiology. If there are any questions, you can ask. So if we do not have any questions, I'll say thank you very much and have a wonderful day.